So yeah, I'm going to go through a little bit about our philosophy around looking for some of these interventions at the beginning, and then summarise one that we've recently launched, and also...
Sorry, I moved them on.
Yeah, okay. And then also one that we've got in the pipeline that we're all pretty excited about, and this is going to be one of the first times we're going to broadcast it out publicly. We're all in the ageing field. We've seen this graph a number of times. Everyone wants to be on the green line. That makes a lot of sense, but the problem is most of the interventions that we talk about are often targeted at the people on the red line, and that makes sense because these are people who know they're sick and they're willing to take an intervention that has some implications to their life, whether it's side effects or just the the responsibility to have to take it every single day. But how do I get the pointer? How do we decide how we get the pointer out? Do we work that out? Is it a clip? Oh, no, that's not what I want to do. Command L, is it? I'm on a Windows now. PC. Control L. Let's try that.
Try actively going. That's all works on the back. So, yeah. Otherwise, it's the cursor on the touchpad. Yeah.
Okay, it's back. Okay, it's working now. Okay, excellent. Okay, so it's made my point for me here. Was it's like if we actually want to be intervening sufficiently early, we need these medicines to be incredibly safe, like virtually no side effects at all, convenient, inexpensive, and also have a way for people to identify someone that will benefit from them, and also track the progress, which is effectively why Juvenescence made the decision relatively early on in our company's life course to both invest in traditional medicines as well as in nutritional interventions that more completely satisfy those criteria. And luckily this point has been made for me by all the last few speakers, is that within our body we typically think about our genes and our proteins, but we're also exposed to hundreds of thousands of very active small molecules. These are byproducts of metabolism. They're also components that actually make up our cells and our organs, and these come from your food, and we nicely just heard from your microbiome, and they circulate through the body and are very important in a number of different phenotypic expressions. The interesting thing that happens in kind of our collective thought is we know about a lot of them, particularly vitamins, because deficiencies in those emerge as quite severe diseases early on in life. What we're realising now is perturbations in some of these emerge as diseases, or at least risk of developing diseases at other life stages as well. And what we like to collectively call these as endoceuticals. So these are effectively medicines that your body either produces itself or gets from its food. I don't think we coined this term. It's what's the one that we're liking to stick to at the moment. And the great thing about these are typically compounds that we've evolved with, and therefore we have a much higher level of their inherent safety. They're also a very defined category of compounds, so the more that we research them, the more we can utilise them in different product solutions that we're looking to bring to market. This is a quick workflow of how we go about this. This is not unique to us, but what you can do now is, because of very large public data sets that have looked at both the genetics and also and analysed some of these metabolites, you can then ask the question of, okay, which of these metabolites are associated with different health outcomes, and then you can go through a quite simple process of picking your preferred option, and then going through the kind of is this going to be effective or not? Run some initial preclinical work, and then go into the clinic. The interesting thing is actually now, because of the computational power that's available is there's actually a number of different ways you can do this in silico, and therefore apply some of the approaches that we heard about earlier around looking at cause influence to see whether these associations we're looking at in these large public data sets are more likely to be causal. To give a confidence in bringing those compounds through the more expensive parts of development. The other side of this is also important as well, that in addition to identifying the compounds, they actually serve as their own biomarkers. So you can both actually look for the populations of people that may be deficient in these, and therefore actively target those people as the groups that will be most likely to respond to them. And it also gives an opportunity to develop the kind of companion tests for people to then track their progress as well. So this is fundamentally at the heart of what we're trying to do with our nutritional interventions. Instead of this, some scientist somewhere has said taking strawberry extract may be beneficial, and then relying on hope that it'll be beneficial to you. We're actually trying to be very precise in terms of looking for these metabolic perturbations that can be restored with some of these small molecules, either directly from produced internally or ones that are from the microbiome, and then restore them in this way. And so this is kind of formed the basis of our discovery efforts, and this is a really nice paper that's come out of a group that we do this with to sort of demonstrate this working in principle. And I won't go through this in a huge amount of detail. I encourage people to look up the paper. It's from a group called 5 Prime in Canada that we work with, and what they did is they effectively looked at all of the different metabolites that are published, their associations with virtually every single age-related disease. We've got Alzheimer's, osteoarthritis, coronary artery disease, and a whole suite of others. And then not only looks at the association with just whether they're high or low, because obviously, as we've heard before, that there's a number of different confounding factors there. What they then went on to do is what we now do with them is you can look at the genetic associations with those metabolites, do what's called a Mendelian randomization study, and then look to see whether those changes are causal in the development of those different diseases. And as you can see, they found several in this preliminary work that we did with them. The one I do want to quickly call out, because it comes out later in my talk, is this association of increased choline catabolism in Alzheimer's being a causal relationship. And this effectively is showing that for every one standard deviation change in this process, you're getting around about a sort of 10% increase in your likelihood of developing Alzheimer's at some stage in your life, which is reasonably significant. But the important thing here is that is just a proof of concept for this whole field. We're really just scratching the surface here. They worked on the metabolome data set, which many of you will be familiar with. It's an industry validated approach from a spin out from a US institute. They test around about 1,500 metabolites, but we now know there's somewhere in the order of 300,000 metabolites which have been characterized in the human. That number is only going up. And the other thing is the technology to both analyze these and then model them is accelerating an exponential process. If you think about this in terms of genetics, we often talk about the human genome being hundreds of thousands. A few years ago, it's now in the hundreds. Same things happens with metabolomics. We're just a little bit earlier in that process. So what we're doing in our group is we're realizing that, and it's probably too much for one group to own that whole process. So what we've started to do is kind of associate ourselves with a network of expert providers we can tap into providers either the best expertise in the analytics or the best expertise in the kind of the processing of that data. And through that, we're building a pipeline of what we believe are going to be this next generation of preventative supplement-stroke medicines based on this methodology. So for the rest of the talk, I'm going to quickly go through a couple of the programs. The first one I'm going to touch on quite quickly, but it relates to the nice little bottle that you've all got in your goodie bags, and that's to do with exogenous ketones. I'll start firstly though with just talking a little bit about Alzheimer's and why I at least believe this is potentially a product with some quite substantial potential. Still the data will come. So we all know about the neuropathology of Alzheimer's. You've seen again, these are two graphs that will not be a surprise to anyone. Alzheimer's typically associated with the build-up of amyloid plaques and tau. You will have seen the graph on the right as well that there's this cascade of pathological features that eventually emerge in the clinical presentation of dementia, but a lot of these different biomarkers predate that. What I want to sort of talk to you about today is there are ones that even predate this that you can start to detect with some of these methods that you can then start to think about intervening in. I broadly called this metabolic dysfunction in that here, but there's probably the more we dig into this, the more precise we can be. There's a third pathological feature that kind of is shown in this graph, in this graphic here, but often isn't talking about quite as openly, is just the shrinkage of the brain. We've talked about all the different things that happen to us throughout our life today, but the other one that is often neglected is our brain shrinks by around about 0.2% a year from around about the age of 30. It's one of those ones we don't really talk about why that is too much. This is something that accelerates in Alzheimer's too. And what I want to sort of talk about is I believe that this is a very good hallmark of a metabolic dysfunction that's happening in the brain, and potentially ketones will be a solution to it. What you see on the left here is this is data from the UK biobanks. If you look at the blood of people and look for levels of different metabolites, what you see is a global elevation in beta-hydroxybutyrate. This is the side product of fat metabolism. But what you also see is a very minimal change in your glucose utilization. This is systemically. What you see when you look in the brain is actually the brain is less glucose on a whole, and this happens again starting kind of sort of the 40, in your age of 40 and progressively accelerates as you get older. And again, this is a process which is even steeper in the dementia. So this leads to the model on the right that there's this gradual energy deficit that sort of develops across life to the point in which you get this emergence of neurodegeneration when you overcome the capacity in the brain. The interesting thing is if you actually then look in both humans, and this is data from mice, is the brain doesn't just try to generate the ketones from the system to feed the brain, but it actually sources them locally. So the brain is around, if you remove all the water from the brain, you have about 50% of it is fats or lipids. This is predominantly in the white matter, and what you see is this is the matter this is the part of the brain that usually gets kind of lost earlier than the rest. And what this study does nicely is and this is as I said in aged mice, is you get this increase in white matter to the point to middle age, and then a steep decline. And an equal association of kind of elevation of ketone bodies, and the hypothesis they drive here is effectively what the body is doing is eating the lipids to fuel the mitochondria to give energy to the brain, and that's why kind of this ability to have ketones will be potentially protective. This doesn't work from us, but actually beat us to it, where they did causal analysis and looking for different genes or genetic mutations that people might have, which make them more likely to produce beta-hydroxybutyrate. They went on and did the causal analysis that we were talking about a second ago, and what you can see nicely on the right is if you look for people over their life course who are able to generate beta-hydroxybutyrate at a slightly higher level, they have higher cognition and likewise a reduced rate of Alzheimer's. So without sort of dwelling on it too much, this led us to developing with our partners at the Buck Institute in California a product which you guys have in your bags there, which is a prodrug or a precursor to BHB, which enables you to get into ketosis without any dietary modifications. We've gone now through 5 clinical studies on either this product or the forerunners to it to pretty categorically show that it gets you into ketosis within about 30 minutes and you stay there for around about 3 hours depending on dose. We don't yet have cognitive data on this, but we're in the process with our collaborators of running a number of different clinical studies, and these will start to report out over the next couple of years. So hopefully in a future one of these meetings we'll have some data to actually go through some of those theories in more detail. I'll segue quickly again just to make sure I stick on time to the main part of this talk, which is plasmalogen restoration. The reason I wanted to start with that ketone story is to put the topic of lipid catabolism, sort of cannibalism in your mind. So lipids, like they're fats. We know they're important. We hear about triglycerides and cholesterol being important to health. But the one thing that I want to stress here is lipids are really, really critical to health. So much so that your brain, as I mentioned earlier, is actually 50% lipids, but the brain is only sort of 2% of your body, and we hear that it takes up about 20% of energy usage. Of that 20% of energy usage, over 25, nearly 30% of that is entirely dedicated to fixing lipid membranes. Because that process is demonstration of how critical maintaining these are to life. And the graph I like is on the right, where it shows that the ability of your lipids to defend against oxidative stress is directly proportional to the longevity of the species. So the fact that your brain is willing to cannibalize lipids to enable the protection of the brain sort of demonstrates how critical that feature is to maintaining our cognition later on in life. So we looked at this and sort of thought, okay, we need to understand this in a little bit more detail. So we looked at the lipidome in aging, and what kept coming up is this odd type of fat called a plasmalogen. And this is just one study of around about 13,000 individuals that looked at various different diseases and their association with metabolites. But trust me, if you look at any of these papers, and there's tens of them, these plasmalogens always come up as one of the ones most negatively associated with the onset of different diseases. The interesting thing is then we did the analysis with our collaborators and demonstrated again that if you have high levels of these plasmalogens, you will typically live around about 5 years longer than people with lower levels of plasmalogens. So you may ask, like, what is a plasmalogen? I think I'll spend a couple of seconds trying to convince you that it's a compound you really should know about. But for some reason, people who have spent many years as a biochemist often have never even stumbled across them once. And in basic, they are just a phospholipids. They make up your cell membrane. In some, depending on the actual tissue, it can be as much as 40%. And what sets them aside from a typical phospholipid is just this fine ether bond here, compared to the normal ester bond you have on a more conventional phospholipid. This change to this bond has a couple of critical features to it that change the conformation of the phospholipid and give it some different properties. Primarily because of the lack of the carbonyl bond, it can pack more tightly, and therefore it's really critical to maintaining the integrity and the stiffness of the membrane. And here's a number of the other features that it's reported to be important in. The ones that I'll flag are protection from oxidization. Again, the vinyl-ether bond works to quench reactive oxygen species to stop the breakdown of the membrane, as well as stabilization of receptors, in particular the respiratory train in the mitochondria as well. I'll briefly talk about the vesicle formation later on in the talk, so I won't touch on that right now. So we worked with a group in the Baker Institute. This is a research institute in Melbourne who specialise in lipidomics, and they have this amazing platform for looking at about 850 different lipid species now. They're one of the leaders in the world. Sorry, again, this is not an easy graph to look at, but what I want to show you here is these blue lines here are either plasmalogens or plasmalogen-like compounds, and what this pattern of having low levels is something that we will repeatedly see in this talk. And what this data set was able to show was, if you look at different populations of people with Alzheimer's, they routinely show low levels of these plasmalogens. The interesting thing is, if you look at the same populations of people across these longitudinal studies, and look for people who had normal cognition at the beginning and then converted to Alzheimer's, these same people had low levels of plasmalogens at the beginning of the study, and what this shows is it's more likely that this low level of plasmalogens is not a kind of an impact of the disease, but it's actually part of the pathology itself. This graph shows this a little bit more nicely with a single plasmalogen. You say the healthy individuals in blue, people with Alzheimer's in red, and then people that convert start lower and then accelerate down during the course of the study. And we then started to think about, okay, so is there anything that we can think about in terms of Alzheimer's? And the one genetic risk factor that often comes up when you talk about Alzheimer's is ApoE. This is the main lipid protein for transporting lipids around the brain, and it's often talked about as the biggest genetic determinant of the disease. What gets a lot of press is the ApoE4 type, because this will have a dramatic increase in your risk of Alzheimer's compared to the what's called the normal E3 allele. The more interesting thing from our perspective is the E2, because whereas most people assume there's not many things we know that can reduce your risk of Alzheimer's, having the ApoE2 gene dramatically does. It can reduce your risk by as much as 60%. When you look within people with the ApoE2 allele, you'll see that these individuals have this pattern of low plasmalogens is entirely inverted in these individuals. And what you can do when you do the kind of analysis of this is the protective kind of treatment effect of the E2 allele appears to be at least 30% driven via the plasmalogen augmentation. So in the real world context, what you effectively see is, this is another bit of data from a larger Alzheimer's study, not by us, is that if you take people in the top 15% of plasmalogens, it will reduce the risk of someone with an A4 allele down to the average of the population. So what I often get asked, and I think I've got a bit of time to go through this, 5 minutes, is the mechanism about how plasmalogens might do this. I won't go through this in a huge amount of detail, but just to put in people's minds that the cholinergic system is really, really critical to your cognition, and this has been known since the early 70s. And then what we didn't necessarily think about at the time is to restore the cholinergic function, the brain will again go to this auto-cannibal sort of it will start to eat its own lipids to produce the choline. What you see with the reduction in the lipids in the cholinergic system is the lipids that get reduced the most are the cholinergic plasmalogens, and that's what this graph is just showing down here. So effectively what you have with the reduction in plasmalogens is you get a reduction in vesicle production, which reduces the acetylcholine signaling and therefore gives you a reduction in cognition. You also get a reduction in the supply of choline, because with the reduction of the vesicles, you also reduce the expression of the choline transporter, which then exacerbates the process more, which causes even more brain shrinkage. I'll skip over this for time, but there's also good data showing that this also results in the cascade of induction of the amyloidogenic process, and also the seeding of tau by increasing the level of cholesterol in the membranes. So you end up getting this theory where low levels of plasmalogens lead to deficiency in the choline signaling, resulting in both the brain shrinkage and cognition that are the key features of Alzheimer's, as well as then kicking off the cascade to result in the more traditional neuropathological hallmarks we all associate with the disease. I'll skip over this, but there are efforts to increase plasmalogens that have been going on for some time now, largely for some orphan diseases, because there are some individuals that are born with severe plasmalogen deficiency. And these do work. They increase peripheral plasmalogens reasonably well, but it's also been known for decades that they don't increase brain plasmalogens. So what we've been doing for the last couple of years is trying to develop a new precursor, and what you can see here is we've been, what we believe, successful, all preliminary data in having some compositions which dramatically increase the plasma exposure and plasmalogens, as well as are the first compounds to increase brain plasmalogens too. And this is a mock-up of our product. And if all things go well, we should be in sort of either early human studies next year or potential market, depending on the route we take. Before I finish, I'm sorry, I've got maybe 30 seconds. I would want to bring it back to the ketosis story and talk about this one study here. And this is a small study which has just been published by our collaborators, and it looked at the ketogenic diet. It's a small, so it's a crossover study in which there are 20 participants on either the ketogenic diet or a kind of a healthy diet that's called the American Heart Association diet. And what they showed is both diets actually did appear to increase cognition, but only the ketogenic diet improved biomarkers of Alzheimer's as well. The really, really important thing for this presentation, sorry to go through it so quickly, is the ketogenic diet. When we did the lipidomics on it, it essentially presented the same phenotype as what you saw in the ApoE2 population. And for us, the important part was the plasmalogens were the lipids that increased the most. And then when you look at the improvements in cognition in individuals, that was directly and significantly proportional to the changes in plasmalogens. So what we're seeing here is evidence that this idea that your body is basically cannibalizing its own lipids to generate enough energy, and then this then leads to this cascade of other cholinergic and then other pathological features. Maybe starting to get some seeds of evidence within this study here. But more to come on this, as I said, as we start to generate our own data. So in summary, we believe this endoceutical approach is probably the only viable way right now to introduce safe preventive medicines that people can take now to prevent disease. Colton Switch is available now. It's I hope a good tasting ketone ester that we can say with confidence will get you into ketosis. And then watch this space, because our plasmalogen product is coming soon, and hopefully the combination of these two will offer some hope for people with higher risk of dementia. So I'll finish there, but here's my thank yous to the group that we work with.
I am conflicted, but that was brilliant, Martin. Absolutely brilliant. We will take a couple of questions. I'm sure there might be one or two. Yes.
Hi, this is very interesting. So how is the level of these plasmalogens sensed in the brain?
How is it sensed?
Yes.
So they're okay. I can actually do this. So down here, there's this enzyme called FAR1, FAR2, And this is both the enzyme which is critical to the production of plasmalogens. It also senses them. So when there's a certain level of plasmalogens met, it effectively turns off the biosynthesis itself. So that's how they say. Or do you mean is there a receptor for them?
Yeah, I was thinking, but that would also do the trick, right? Because then it would kind of see the level itself, right? Because normally lipids need to be equilibrated, right?
Yes, they're auto-sensing. So they get to a certain level and then it turns off their biosynthesis. Exactly.
Yeah.
And it does through that FAR1, FAR2 gene.
But since you mentioned receptors, are there receptors for it?
Pardon?
Since you mentioned receptors, are there receptors for it?
I don't know if there's specific receptors that initiate signaling from them. There are various different GPCRs that people have shown have some activity when they're exposed to circulating plasmalogens. The difficulty with those is you very rarely get free circulating plasmalogens. They're often in these lipoprotein particles, so I don't know how much you can actually believe some of that data.
Thank you.
A hand down the front here.
Hi, great talk. And what we do with age-related macular degeneration as a charity, we also try to go to the same route. But we always come with a problem which is, it is a neurodegenerative condition, many of this you're looking at. It takes a long time to develop. How do you track whether your compounds or your treatments are having an effect? Do you have any sort of surrogate marker that does depend on blood drawing, for instance, right?
For measuring cognition and changes in neurodegeneration.
Correct. So
We don't have anything specific. We're somewhat relying on the fact that the drug companies have been developing many of these to support their amyloid products over the last few years, and so there are some good surrogate markers now for those mechanisms, and we haven't got direct data showing that the plasmalogens will modify those just yet, but that's going to be something we're going to be looking at in the clinical work we have planned for the next couple of years. The good thing is from blood draws with these, you don't need to have a venal blood draw. You can measure these just from a dry blood spot, and we've got that technology measured. It's relatively well developed at this stage, and you can measure all of these ether lipids that way.
is cognitive detection too late, actually? Another question. Sorry.
Probably yes. Yeah, it's probably like we're developing these for something you can take intentionally early on in life to prevent the decline. We have this debate a lot about whether that is the right approach or whether we should be trying to look at interventions that for people who've already got the cognitive decline or not. But yeah, more to come on that.
Last quick question. Thank you.
Thank you, brilliant talk. Do you know the effect of statins on the plasmalogens? Because cognitive decline for people who take statins is confirmed at the moment at the level of mitochondria. So how statin can increase lowering?
Yeah, so we actually asked this question of Peter, who I've presented this work. It's mostly a collaborators' work. So sorry if I did put my foot in my mouth a couple of times. There is some reports that statins do have a modulatory effect on plasmalogens, but it doesn't seem to be anything substantial.
Brilliant, Martin. I'm going to end it there. Thank you very much, Martin. Cheers for your.